My name is Mhairi Morris, and I'm a senior lecturer here at Loughborough University. I work in the School of Sport, Exercise and Health Sciences, and we do everything from exercise physiology through to molecular biosciences. I myself work on breast cancer and exercise. So what we're currently trying to do is build a 3D model of the obesogenic breast cancer microenvironment, and we're incorporating fat cells from two different sources. So we have fat cells from visceral fat, which is your bad fat that you have around the belly, and then we have fat cells from subcutaneous fat, which is your good fat, if you like, that burns energy. And what we want to look at is whether exercise has a differential effect on the cancer cells, whether they are incubated with either the visceral fat or the subcutaneous fat. 3D models are much more representative of what's happening in vivo than 2D models, and I liken it to, you can play a game of football lying down on the ground side by side and not being able to move, but it's not very representative of a real game of football. And it's the same with cells. When you grow them in 2D cultures, they only interact side by side, whereas when you culture them in 3 dimensions, they're able to interact on all 3 dimensions with each other, but also you can incorporate other cell types into that model and model these interactions with other cells, extracellular matrix that they would have in that natural tumour microenvironment, and build something that is a little bit more, or a lot more, representative of what happens in vivo. Primarily I use CellTiter Glo 3D to look at cell viability, and more recently I've also been exploring the use of the Lumit 67 kit to look at proliferation as well. CellTiter Glo 3D is a lytic assay. So you add it to your wells and it lyses the cells, they release ATP, and that ATP is then used in the luciferase reaction, which produces a luminescent signal, and that signal is proportionate to the number of metabolically active or viable cells in your well. I used to use the MTT assay to measure viability, and then latterly the WIST1 assay. But the trouble we had was when we moved from 2D to 3D, we couldn't replicate the results every time. And then I was introduced to CellTiter Glo 3D, and it's been wonderful actually, because it's every time I get the reproducible result that I can go from batch to batch, experiment to experiment, and I never had that reproducibility before with the other assays. It was really easy to adopt this assay because it is so simple to use, and it didn't require much in the way of optimization beyond your basic project-specific optimization. So you simply add it to each well, pop it on a plate shaker for 5 minutes, and then incubate it in the dark for 25 minutes, and then read it, and then you get your results and off you go. One of the key questions that we are asking is, how does exercise impact on cell viability? So it's really critical for us to have an accurate, reproducible measure of that viability every time. So one of the big benefits of CellTiter Glo 3D is time saving, actually. With other assays like MTT, you've got a 4-hour incubation. With CellTiter Glo 3D, it's half an hour and you're done and dusted. So think about what your time is worth and how much time you spend in the lab. It can really accelerate the results that you get and maximise your efficiency. One of the unexpected benefits of using CellTiter Glo 3D is that you don't have to digest your matrix before running the assay. So I culture my spheroids and my organoids in a matrix, and with other assays you would have to do several steps to digest that matrix before you can even go on to run the assay. CellTiter Glo, you just pop it on and off you go. So when we culture our cancer cells with the fat cells, we actually see enhanced viability. But when we overlay exercise on top of that, we actually see a reduction in viability. So really interestingly, what we're finding is that exercise can maybe negate some of these negative effects that fat cells have on cancer in the tumour microenvironment. Our next steps really, now that we've built the model, is to use that model to start unpicking some of the mechanistic details underlying the effects that we see with exercise in cancer.